So you'll be introducing.
No, so
Okay.
Mike's still our next meeting, so I just wanted to know who's left.
Okay.
Sit there or don't sit there.
No.
That's where the cat comes.
Here, everybody who's here in person, and thanks for being here, people. So I just want to welcome everybody and acknowledge this Medical Scholar Seminar Series. By way of reminder, Timothy Johnson, founding faculty of the Tech School of Medicine, and he was dedicated interface between research, science, and medicine, and making sure that there's a good forum to bring those things together. So with this passing after 2 years that I started, we made this program and several it's an outstanding program in many respects. Very unique. Physician scientists here were very active. World-class speakers here today, and I have the honor of introducing the introducer, Dr. Anthony Malatya from the Hedgehog Research Institute, who will introduce
It really is a great pleasure and honor to meet you. We're also
Yeah, go ahead and turn yours on. The microphone is. Well, we couldn't hear you there. Yeah, keep that one on. Yep, because then we can hear you there.
Okay.
Yeah, we're going to be doubling it up. Sorry about that.
Okay, well, thank you so much all for coming to hear my talk. I thought, this is my first talk. So, contact me and said, send some papers. So, I said, I'm coming back. They're going to want to talk, interact. So, and I hope one of the things we talked about there, you might think that careers are kind of linear, they start at one point and singular path. It's not the case. Going one way or another, it depends on that simplify thinking about what I think applies to all of us.
susceptibility. So why is that? What is the nature of susceptibility to cancer, and which are the cells that must have unique properties in the developing brain that render themselves to be susceptible oncogenesis? So that was the journey that I began on. And I decided that I would study the second one down, medulloblastomas. They were called at the time primitive neuroectodermal tumors. Or their developmental origin was kind of assumed by the name. And it made sense that was probably the case because they look very much like cerebellar granule. They occur in the cerebellum, and they look very much like cerebellar granule cells. So I thought, well, it's probably a tumor of cerebellar granule cells. That's an interesting paradigm to think about. This just kind of gives the perspective of, how common are medulloblastomas. We used to say they accounted for about 20% of all pediatric brain tumors, and we no longer say that, in part because as our precision has grown and we understand things at a much better level. In the old days, 30 years ago, all we had to go with was microscopes. We'd look under the microscope and we'd know where the tumor was, and we'd say, well, this is a medulloblastoma. As time goes on, we came to realize that, well, some of those tumors are actually a different tumor called an atypical teratoid rhabdoid tumor, which was important. And they're actually very different. They have a different cell of origin. They have a different mechanism of oncogenesis, but most importantly, they have a horrific prognosis. So once we define that atypical teratoid rhabdoid tumors are no longer called medulloblastomas, the prognosis of medulloblastomas dramatically improved because we removed this type of tumor that was actually a very poor prognosis tumor. So the value of precision of diagnosis at a molecular level and sometimes cellular level was really illustrated in the 1990s with that discovery, and more to be said about that. But now I can say, well, medulloblastomas really are only a little less than 10%, not 20%, like we used to say. Still lots of work to be done on gliomas and other tumors, but this is where I've focused my energy over the last 30 years. This is what one looks like. Here's an MRI scan, sagittal section. It occurs by definition in and around the cerebellum. And these tumors they always occur in this location. They have a tendency to spread throughout the nervous system. About a third of them present with distant metastasis up and down the neuraxis. Occasionally, they even escape the neuraxis and go to other organs. And because of that tendency, they're very aggressively treated. They have, after surgical excision for greater than 3-year-olds, because of data below, we give radiation to the entire brain and spine and then multi-drug chemotherapy for about a year. And for standard risk patients, for patients who are greater than 3 years old, who have no evidence of metastasis and had a good resection, using that approach, there's about an 80% survival, which is not horrific. It's not, you would like it to be better. High risk, meaning with metastases a little bit lower. But this graph really begins to tell the rest of the story, which is if you irradiate the brain of a child below the age of 5, it's pretty devastating. They have an IQ drop if you follow them over time of about 40 points. And if you put it into practical terms, you follow them out into life, how many of these kids go on and live independently, have a job and a family and a home, a life living on their own? It's a very, very small percentage. Most of them live in some sort of setting that requires supervision and a job that is no way self-supporting. And they also have a high incidence of hearing loss. They get an ototoxic drug, cisplatin, plus radiation to the ear, so a high incidence of having hearing aids and substantial hearing loss. They have shortened truncal. If you irradiate a growing spinal column, it gets short, so they have a very characteristic short trunk with long arms and so forth. So the treatments are effective in terms of survival, but the quality of life is substantially degraded. And so our objectives are not just to try to improve survival, but we also want to improve the quality of life of those who survive. So we began with this model of thinking of, well, these are probably cerebellar granule cell tumors. Maybe we can just map them out onto the progress of granule cell development. Granule cells proliferate on the peel surface of the cerebellum, drop out of the cell cycle, migrate down. So here's the EGL, migrating cells past the Purkinje cell layer to form the internal granule cell layer. And there's a differential expression of TREC receptors in early versus late granule cell migration. And we showed data, this was back from the early 90s, that TREC-C, the marker of most differentiated granule cells, is associated with a good prognosis. So we wondered, what does that mean in the landscape? How important is that? Is that like a defining feature that really overshadows all other features of the tumors? Or it's hard to know what to make of it in terms of the relative importance of that, and then what underlies that. So starting around 1996, 7, 8, I started to get interested in trying to do transcriptional profiling. And these are early days, so there was very little available to do it until one morning at a coffee and donut meet and greet with other young investigators. I ran into Todd Gollum, who was another young guy. He was a couple years behind me, but he had set up a program with Eric Lander to do transcriptional profiling and worked out with Affymetrix. So we got in on the ground floor of Affymetrix, and he had hired, and he had a platform, but he needed projects. And I had a project looking for a platform. So it was just a really, you go to these coffee meet and greets and you're not sure what you're going to get out of them. That was a life-changing event for me. And for him as well, because I was one of the first projects that was really successful for him. And not only did I get access to the platform, but he had hired biostatisticians and informatics out of IBM and out of these physicists and people who had no knowledge in particular about biology. And so I could sit with them, and as a biologist who understood the issues, and over the course of years, we developed the methods that became the methods used in genomic analysis. I was on the team that developed gene set enrichment analysis, if you know what that is. I'm an author on the original paper. So it was a conjunction of across disciplines of a moment that I couldn't have possibly anticipated, that I said, I'm there, I can see the value. And that's really, that was a game changer as well. So we started, first, we had no idea how discriminating this method was. So we took a group of malignant tumors and we got expression profiles using Affymetrix microarrays. And first just asked, can you tell one from the next by transcription profiling? And the answer was yes. You can see this, each column's a different tumor, each row's a different gene. High expression is red, low is blue. They each have a different transcription signature. And if you then use that transcription signatures and map them out in three-dimensional gene expression space, these are the top three principal components in principal component analysis, you can see that they tend to cluster together. So medulloblastomas, gliomas, rhabdoid tumors all cluster in the same way, same region of space, and even rhabdoid tumors from different parts of the body, brain versus kidneys, others, they all tend to lump in the same space. And this group of tumors that is called PNETs, we're kind of in between all these different groups. And these PNETs occur in different parts of the brain. And we had pretty skeptical about them as a class at the time because they seemed like they had behavior that was all over the map. They were very, sometimes it was hard to tell whether they were going to behave like a high-grade glioma and be highly invasive of the brain versus more like a medulloblastoma and spread all over the place. And the difference of treatment is dramatic For gliomas, you irradiate just part of the brain. For medulloblastomas, you do the whole brain and spine. So it's for treatment. And the consequences of that to the child are all or none. If you irradiate like glioma and it spreads all over the place, game over. There's no way that you're going to be able to go back and treat it. So it turns out those PNETs turn out to be a whole bunch of different types of tumors. This isn't my data. This came out, Dominic Sturm and the Heidelberg group showed that if you get a large number of them, a number of them actually are various forms of glioma. But there were also other strange things. This embryonal tumor with multi-layered rosettes, a tumor I'll talk about briefly at the end of the talk. Various neuroblastomas and other things. So these tumors that we called PNETs, not in the cerebellum, the data from 2002 showed that medulloblastomas are different than other PNETs, different class. And this data now shows that PNETs as a class actually break down to a bunch of other different tumors. So we've eliminated, the World Health Organization eliminated the term primitive neuroectodermal tumor from the lexicon, which to you guys doesn't mean anything, but for a person who's been in the field for many years, that was a very common term that we no longer use. So that's that, and it showed the increasing accuracy that we were able to make a diagnosis now that we have molecular classification. When we, as I said, medulloblastomas, we knew were heterogeneous. Sometimes they very much respond to treatment, sometimes they don't. They all look the same under the microscope. When we started applying these genomic methods to medulloblastomas, and then using methods, in this case, non-negative matrix factorization, which looks at a data set and then breaks it down into the optimal number of common subtypes based on a metagene that it calculates. So you can define statistically what's the optimal number. You can see that for medulloblastomas, they kind of break down into 4 broad groups. There's this one here, and this one here are pretty tight. These two are a little bit less tight. You can see there's heterogeneity within this, and it looks like it might be there as well. So we, with this data and with other colleagues collecting similar data, came to an international consensus that there were 4 principal subtypes based on transcription profile. And that consensus that was reached in 2012 continues to this day. When we looked at the patterns of expression using gene set enrichment analysis, the method that we had developed, this C3 group turned out to have a very strong sonic hedgehog signature. The C6 group had a Wnt signature, and the C5C1, which we call group 3 now, has had a strong mixed signature. So the mechanism driving these different groups was evident within the transcriptional profile. When we looked at the DNA copy number, so this is now SNP array data, it's a DNA copy number from SNP arrays that are profiled by chromosome. You can see just to the eye, you don't need a statistical test, although if you did, they're very different from one another, that each of these transcription-defined subtypes really are very different from one another genetically. And in fact, the sonic hedgehog subtype here has a very common loss of the sonic hedgehog receptor locus on chromosome 9, which is consistent with the mechanisms, which we'll talk about in a little bit, of how to activate the sonic hedgehog pathway. And this Wnt, or this MYC signature subtype, often has gain of copy number or overt amplification of MYC behind it, driving the expression of MYC. So again, at a DNA level and RNA level, a clear idea of what might be behind these. And finally, when, as I said, an international consensus was reached, and this now is accepted by the World Health Organization in 2016 as part of the pathologic classification of medulloblastomas, really breaks down into 4 broad groups. The Wnt group, driven by the Wnt pathway, is distinguished by having an excellent prognosis, 95% survival, whereas this group 3 with MYC amplification or MYC overexpression has a relatively poor prognosis. So we know now, and we knew then, that the reason some tumors responded to treatment and others didn't, although they looked the same, was because they really were just different tumors. They had different molecular makeup, different mechanisms driving them. And we know now, in some cases, different cells of origin. They're derived from a different type of cell in developing brain. They have differences in age distribution. It's interesting that group 3 and group 4 are much more common in males than females. We don't know why. That's one of the mysteries that we still to be solved. Different age distributions. And the sonic hedgehog group is interesting in that there's a high incidence in babies, infants. It drops and then it increases again in adults. And if you think of this in developmental terms, hard to explain why that might be. Probably different populations of cells. And I think that the infant tumors actually stand out in ways that they have a uniquely good prognosis compared to the others, and in many cases respond to chemotherapy only, no radiation required, which is a great boon. So like precision, that's beyond what I've presented, but that's the more recent data that's come out. So the molecular groups really define the heterogeneity of the tumors and a big insight and a total change in how we think about these tumors. So just to remind you about the hedgehog pathway and its importance in granule cell development. Granule cells begin a journey starting in the ventricular zone where they cross the rhombic lip and enter into to form the EGL. And if you activate sonic hedgehog down in this location, they don't form tumors. It's only when they cross the rhombic lip and become committed progenitors, if you activate the sonic hedgehog pathway, then in an uncontrolled way, those cells form tumors. So it tells you that the developmental state of the cell is critically important, very likely because prior to crossing the rhombic lip, they just don't have the mechanism to respond to sonic hedgehog. It's just not there. And it takes a step of development for them to be poised for that to happen. And there's good evidence now that the Wnt subtype tumors are derived from the lower rhombic lip. They're still from cells within the brainstem that are driven by the Wnt pathway. And they then lose that susceptibility when they cross and they're multi-potential, so they go in different directions. But those are the cells that are basis of the Wnt tumors. So sonic hedgehog in granule cell development is an important molecule that is secreted by Purkinje cells. It's really probably not just a mitogen. It kind of is involved in the regulation of the development of the cells. They proliferate during the time that they're responsive to sonic hedgehog, leave the cell cycle and migrate past the Purkinje cell layer to form the IGL, as I said. And the mechanism is still being worked. This doesn't begin. This is a very old slide here because there's a very interesting mechanism about sonic hedgehog signaling that involves primary cilia, and it's a story in of itself. And I should change my slide now that I'm saying this because it's such a great story. But this is very simplistic, and it makes the point that this hedgehog receptor patched in its unbound state tonically inhibits another transmembrane protein, smoothened, and it's in binding of hedgehog, then it blocks that inhibition and activates smoothened as a result. And if you then knock out patched, either by mutation of the gene or loss of the locus of the patched receptor so that smoothened is uninhibited by patched, then it just remains constitutively active. And if you do this and if you knock out patched, make heterozygous mice with patched knockout, you find that these mice have rests of proliferating granule cells, about a fifth of which go on to form tumors. So there's probably a second or third genetic hit or epigenetic hit that occurs that takes it out of that cell cycle state and into more of a tumorigenic state. And you might imagine then that if you could only block, in this case, block smoothened, you might stop the tumors. And that's the case. There are drugs that are now that block smoothened as part of the sonic hedgehog pathway inhibiting process. And those tumors do stop growing if you block smoothened, as long as the receptor doesn't mutate and become, or the protein doesn't mutate and become insensitive to the drug. But unfortunately, in children, a pretty high percentage of sonic hedgehog tumors have activating mutations downstream of smoothened. And so those tumors, as predicted, won't respond to smoothened inhibitors. And so that smoothened blockers are of no value for them. And it also turns out that smoothened is in the hedgehog pathway is important for other parts of the body, like bones. So if you block smoothened in a child, it blocks their growth plates. And so it turns out that smoothened inhibition is not an effective strategy for medulloblastoma treatment yet, although yet more to come. But our parsing tumors into a sonic hedgehog subtype has yet to fundamentally change how we treat the tumors. We still treat them with radiation and chemotherapy. When we then looked at the somatic mutations of the tumors, this was work that came out a year or two later, we and others found that medulloblastomas and many childhood tumors have a very low mutation burden. This is a log scale. So melanoma has 100-fold higher number of somatic mutations as compared to medulloblastoma. So one fundamental feature of childhood cancers is the relative rate of somatic mutation is quite low, which has lots of other implications that we won't go into, but that's a fact. But if you then map those mutations onto the subtypes, just like we did with DNA copy number, the top part of this table is copy number. I like to say this is like 10 years of my life in one PowerPoint slide. The mutations map as you would expect. Mutations of the sonic hedgehog pathway are really segregated only to the sonic hedgehog subtype. For the Wnt pathway, a high incidence of mutation of beta-catenin, the transcription factor. It's a mutation that stabilizes beta-catenin. And interestingly, these other tumors really have a very low incidence of recurring somatic mutation. They're really driven more by copy number, and the details of that are being worked out. But segregation by subtype is the name of the game. And what about all those other, as I showed you in the previous, there's a common mutation of beta-catenin and a couple of other genes in the Wnt subtype. But if you look across all the tumors, there's nothing that really stands out as a gene that's frequently mutated. It's many different genes. But if we then map them as a class onto various mechanisms, it turned out that many of the hits hit transcription regulating processes. So instead, and this was another fundamental change in our thinking that came out of this data. Up until this point, I had envisioned, and most of us envisioned, the money of treating cancer was going to be receptor tyrosine kinases or the MAP kinase pathway or RAS or SART, whatever, genes that are converging signals into the nucleus. And the scary news that came out in 2011 and 12 is that the mutations are largely in the nucleus. It's genes that are involved in the chromatin unwinding, or that histone marks, things that are involved in actual gene transcription regulation. So a whole new level of complexity came up in terms of thinking about how do we approach this problem came from that. As I said, in the 1990s, another type of tumor, this atypical teratoid rhabdoid tumor, came to light. And they tend to occur in very young children, 1 to 3 years. They're treated very aggressively, median survival 1 year. I mean, and it's not uncommon for series to have no survivors. It's a terrible tumor. Getting a little bit better these days. And the one gene that's mutated, so the gene had been identified in the 1990s, and we worked with Charlie Roberts' group to show that unlike medulloblastomas that have lots of copy number gains and losses, that doesn't really happen in rhabdoid tumors. And in fact, the only locus that was consistently lost in this gistic plot is SNF5, which is a component of the switch SNF complex. So another example of a fundamental gene transcription regulating gene that, in this case, the only recurring mutation leads to this very, very malignant tumor. So again, highly malignant childhood brain tumors, low level of mutation, but fundamental disruption of signal transduction, of gene transcription regulation. So it's part of switch SNF. It's a loss of function mutation, and in trying to, the cell of origin still has not been fully worked out, but it's known that it only forms a tumor in very restricted cell lineages for a very limited period of time. That's why one mutation of a gene doesn't cause cancer constantly, because fortunately in this case, it has to be a very narrow window for that in the development of one restricted number of cells for that mutation to form a cancer. Another horrific childhood brain tumor, diffuse intrinsic Pontine glioma, now known as diffuse midline glioma. These kids are a little bit older, 5 to 10 years. Here's one right here. It's within the brainstem itself. So you can see it's just kind of intercalated within. In fact, it's kind of growing around the basilar artery right here. So there's no way you can't approach these surgically. You would have, obviously. In fact, we frequently don't even biopsy, and we go only by the MRI image and say, this is DIPG. They get radiation, they get a little bit better, median survival 9 months. So it's a terrible tumor. And it turned out when this was not my data, this was others, the gene that's most frequently mutated in this tumor is histone H3.3, a histone mark. And this is a mark, it's a very interesting mark. It's, and probably you've run across this one. When it's trimethylated versus acetylated, it's like an on-off switch for differentiation. So what happens if you mutate this gene through EZH2, somehow it leads to a hypomethylation of the genome with hypermethylation of specific genes, a set of genes that appear to be different genes of differentiation. And basically what this does is it locks the cell into an undifferentiated state. So instead, it disables the mechanism of differentiation to mutate this gene. And for some reason, again, in this, it's thought now to be myelinating oligodendrocytes. It forms this terrible cancer. It only occurs, and it's really the only cancer in which this mutation occurs on a regular basis. And it tends to occur in the midline, in the brainstem or the diencephalon. And it occurs at specific ages between, usually in the first decade, sometimes into the second decade. As I said, it's thought to be an activating mutation of myelinating oligodendrocytes. And these are oligodendrocytes that are presumably myelinating the corticospinal tract during the early development of the brain. And once that myelination and developmental stage is finished, those one would have to assume that these mutations are stochastic. They occur all the time in different genes throughout the genome. And it's only the state of the cell that leads to cancer when there's a concurrence of the wrong developmental state with the wrong mutation. And so that's what is behind these tumors. And unfortunately, even though we now have known this for 10 years, we've yet to improve the outcome of this terrible tumor, but many people are working on it. And then finally, another really malignant tumor, ETMR, I referred to. We used to think of this as one of the many tumors that make up PNETs. Again, they occur very early, 2 years, median survival 1 year. And they're distinguished by having an amplification of a microRNA cluster on chromosome 19, C19MC, that is amplified as a fusion with the promoter of a chloride channel, TTYH1. And when that occurs, it leads to the constitutive expression of this microRNA cluster, which through a series of steps leads to a loss of inhibition of a DNA methylase that normally is only expressed during development, and the consequences of which is again, it kind of pushes it into an embryonic state, a poorly differentiated state. So the story that is emerging from all this is that that for pediatric cancers, this notion of signal transduction pathways being aberrantly activated is changing, a paradigm's changing, and we don't think of cancer as only being a disorder of excessive proliferation or loss of apoptosis. It's really a state of differentiation disease, and I think that's a critical component. And importantly, you could say, well, let's give a drug to activate differentiation if that's the case. It wouldn't work because you've disabled the mechanism of differentiation unless you could find another way to work around that mechanistically. So as I said, it's not just a disorder of excessive proliferation, it's a disorder of differentiation. And that's just the reality of where we're going. And understanding the regulation of the epigenome is now become a really important part of our thinking about where do we go from here in terms of developing cancer therapies. We can target specific components of it, and that's being tried. So far, the drugs haven't made a big difference. I'm sure because it's, in the end, more complicated than what we even think now. We've tried another approach, and I just mentioned this because I thought it was pretty cool set of experiments. We looked at the proteome. This was published a couple of years ago now, and we worked with Steve Carr at the Broad, and he'd worked out a process of probing the phosphoproteome he has like 15,000 phosphomotifs that he can map. And so you can tell from his data whether specific tyrosines or serines or threonines are on specific molecules are phosphorylated or not. Very cool data. And so we applied some of the analysis methods that we have been working on, identified a protein PRKDC as a kinase that looked pretty interesting in one of the subtypes. And we thought it was interesting because PRKDC actually phosphorylates a serine on MYC that stabilizes MYC. So if you have upregulated PRKDC, it may be one of the things that's stabilizing MYC, and MYC is an oncogene that drives proliferation, especially if in the normal processing of MYC, which is in a very regulated state. You don't want to have transcription factors out there kind of doing their thing unless you keep them under tight control. So normally threonine 58 gets phosphorylated and kind of sends them down. They get ubiquitinated and then undergo proteolysis on that way. So we found examples of MYC that had both serine 62 and threonine 58 phosphorylated, that over phosphorylation of 62. We thought maybe if we block PRKDC, we could fundamentally change the biology of the tumors. And in a cell-based model, we showed that you can increase the radiation sensitivity by blocking PRKDC. So we and others are looking for various Achilles tendons, if we can find them within these tumors, that may be approachable by therapies. Unfortunately, there's not a PRKDC drug right now, so even though we can show it in a dish, we can't show it in a human. So where are we today? Well, we have not gotten to the point yet of replacing radiation and chemotherapy with targeted therapies. And so the era of precision medicine doesn't occur in a single step. It's not like one day you cross a threshold, turn off the radiation machines and stop giving chemotherapy and replace it with giving a pill. But it's a stepwise process, and we've made substantial progress. If you think about it, I said this at lunch, you know, if you had an individual child, if your child came in down with a malignant tumor and somebody came to you and said, well, I have this treatment that's 80% effective, it has a lot of side effects, but I have a pill, so I recommend you not give radiation and just take this pill. We're not sure if the pill is going to work or not. You would never, first of all, you'd never suggest that. That would be completely unethical. Two, nobody in their right mind would agree to it. It's the wrong way to go. It will be a stepwise process of saving the survival benefits that you currently have, dialing back the current therapies as fast as you can dial them. If you know, amazing, if a Wnt drug came along and it was really, really effective, you might say, let's not give radiation and give it a try because you haven't burned any bridges. And then if it fails, then you give radiation or something like that, maybe. But that's not yet where we are. And we're not at the point of having that decision making. So where we are is we've identified Wnt as a particularly good prognosis subtype. So if a child now is diagnosed with this subtype, we can say at the time of diagnosis, you've got a 95% chance of surviving this. And we can now lower the radiation dose to sometimes less than half of what the original radiation dose, which makes the difference. So we have improved the quality of life, and we can also lower the amount of chemotherapy so that they won't have peripheral neuropathy and some of the other complications of chemotherapy. So we can preserve survival and improve the quality of life. In the case of the sonic hedgehog subtype, as I've already said, unfortunately, the inhibitors so far have not been useful, but we've identified a particularly poor risk subtype that occurs with p53 mutation. P53 is a DNA repair gene. And so in this case, the prognosis of these tumors is so poor, you might say giving radiation may be making matters worse. You know, maybe we should think about other ways of treating them. Or many of the drugs that we use are DNA alkylating agents. They mutate DNA. They act by mutating DNA and triggering apoptosis. In this case, probably isn't helpful. So maybe we should go down the path of using methotrexate or something that doesn't cause DNA mutation. So it changes our thinking about how to approach current therapies. And it also gives us much greater precision in terms of prognosis and dialing up and down radiation. We've also gotten much better at focusing radiation for the boost doses. And so we've made a lot of progress in improving quality of life, of shaping our current therapies and changing the mindset of people around the world, really, about what's important about these tumors. And when I was doing research, really up until probably 2010, which is not that long ago, I think my clinical colleagues kind of were amused by me and thought I did research because it was my hobby and I like to write papers and grants and things like that, but it had no value to the care of patients. They no longer say that. In fact, the conference calls in 2011 and 12 totally changed. They got it. They could see the value of this and where it was going. And World Health Organization, as I said, in 2016, changed the diagnosis of the tumors. You cannot diagnose a tumor without the molecular classification. So we've made progress, but we have a long way to go. And I'll end by, here's colleagues that I've worked with, but I always like to say this to I interview a lot of resident applications, and I say to them, you know, my generation took a bunch of diseases with funny names. We didn't understand them, Wernicke-Hoffman disease, other things like that. And we got them to the point of where we understand them at a pretty basic level. And we're starting to see some major wins, like spinal muscular atrophy. It used to be Wernicke-Hoffman disease. Now it's a disease of progressive muscle weakness, and the children who have it in its worst forms have 100% mortality. We now have a treatment, a genetic-based treatment, antisense oligonucleotides, whose children are now walking from 100% mortality to walking. So the challenge to you, to your generation, the students in the room, is my generation got us to this point. Now it's your job is to fix the problem. You got pressures on. A couple of decades to work on this. And as I said this morning, I think 30 years from now, when you're standing up here talking to the students who are just getting started, you're going to have amazing stories to tell them of the things that have happened in your career that were unthinkable when you got started. So thank you for your interest and time, and be happy to answer any questions you might have.
So we'll take questions from the room, but we also have some online questions that I will read and stop. Start. Question all the way up.
So single cell analysis is underway and it's being done. It's still at the level of trying to understand the basic biology of the tumor, not yet quite at the level of making therapeutic decisions. And we're defining a level of heterogeneity at a cellular level that we didn't see at this bulk expression level. That no surprise. And there are, interesting things that come out of that type of analysis. So that we've hypothesized that there's a cancer stem cell. That cancer is actually, when you look at a cancer, there's a very small population of cells that really are the core drivers of growth of the tumor, and then they kind of differentiate. And most of the cells that you see are kind of downstream of that. And so, if you were to isolate the cells within a tumor and transplant most of the cells into an animal, they wouldn't form tumors. But if you were able to identify the stem cells, they would. So they're very different. And the single cell data supports that conclusion as well. And we've kind of known it biologically. But so we're beginning to understand at a fundamental level what the ecosystem within the tumor is like, but it hasn't resulted in a therapeutic difference yet. Yeah, I'm sure so can tumors begin in utero? And the answer is yes. And many of them are horrific tumors and not necessarily even typable at this point. If a child is born with a massively malignant tumor, sometimes it's not even treated. It's just, it's too far. But I think that the idea of a truth that you're referring to is that there's not just a single hit. There's something that gets things started, and then we've known for many years now there's a second hit or a third hit. There's another alteration that occurs over time, And so there's an evolution that occurs so if it may begin in utero or may not, or may occur during the first year of life, but the tumor doesn't actually become evident until age 10, implies that there are things that are happening between that initiation hit and the formal emergence of tumor. And it's sometimes it's obvious it's a second hit, a second genetic hit. In other cases, it's not so clear that it's genetic. It could be epigenetic or something, a modifier of some sort that alters it. So the evolution of tumors over time is an important concept that we're still learning about.
First of all, thank you. Fantastic. I'm really intrigued by the sonic hedgehog group and the bimodal distribution of age. And I'm just wondering what you think of about the later onset one. So is it just a slow ride for those cells to de-differentiation, or back to what you just said, in terms of the second hit?
It may be just the accrual of genetic damage, just like we were showing in the mutations of children cancer tend to be simpler than those of adults. It may be that there's kind of a latent group of precancerous cells that then become cancerous once they accumulate enough genetic damage. That might be one possibility. Otherwise, unless there's a sonic hedgehog driven population of cells that emerges later on, which I don't know about, then that might explain it. But I tend to think it's latency rather than a different population of cells.
So we have two questions from online. First one I believe is from Metroblastomas, and he asks, immunotherapies have demonstrated success in treating many different malignancies. We struggle to show is looking at the CNS neoplasms. Do you think immunotherapies could eventually be an effective treatment for CNS malignancies? And if so, what are some of the barriers preventing immunotherapies from showing success?
Yeah, so an early truism seems to be that the degree of genetic abnormality is important. And so pediatric tumors are not as genetically abnormal as adult tumors. So melanomas, which were, the immune therapies have been game changing. And whoever would have thought that metastatic melanoma would be a treatable disease. And their mutation rate is extremely high because they're getting bombarded with, UV all the time. So one is the basic substrate. There's just the immune. They're not sufficiently. Their immune evasion mechanisms are not developed at a level where blocking them makes a different PD-1 inhibitors, or that their unique antigenic properties are not so different. That it makes it different. That those are possibilities. Another thing is the blood-brain barrier, and trying to get complex treatments across the blood-brain barrier into a tumor does make a difference. And so it hasn't so far for any CNS, and really for any CNS tumor, the immune-based therapies have not been successful. However, for years, people have dabbled in immune-based therapies for decades, and to the point where people were going, "Oh, is anything ever going to come of this?" And by golly, it's been a game changer. So don't count it out yet. Maybe it'll happen.
So another medical student, Samantha Howard, first says, "Thank you for a fantastic talk." Echo that. I understand that a common characteristic of fully differentiated cancer cell is the ability to more and less differentiated states. Are there epigenetic features that would promote that plasticity? Blocking the cellular.
Yeah, I think normally there are mechanisms that can move an undifferentiated cell to a more differentiated state, and there are examples in cancer of differentiation-based therapies. It's just that if the basic mechanism, it's like apoptosis. If p53 is out of commission, it's just not going to work, because you've basically you've derailed one of the main pathways by which apoptosis is activated. So the nature of cancer is, in some cases, the very thing that you would like to do no longer works, and that which makes it so hard to figure out a way to get around.
First, is there a gender difference between different molecular subtypes of Metroblastomas? And second, he asks, is there any evidence from single cell sequencing showing that there is intratumoral heterogeneity in a given Metroblastoma? If so, how will this heterogeneity impact?
So yes, there are in the so-called group 3 and group 4 subtypes, there's a very strong male predominance, 2 to 1, that in some cases 3 to 1, higher incidence in males over females. They have they have this very common genetic abnormality, isochromosome 17q, which is a loss, it's a duplication of Q and a loss of one arm of P, if I recall. And no one really knows what that's all about. I think you lose one copy of P53 with that. But what is the gene of isochromosome 17q? Nobody really knows. There's lots of theories about it. But it is associated with this imbalance of male versus female. You wonder if there's some, X chromosome, Y chromosome, what is the difference? Unknown. But it's an interesting problem. And we've thought about ways to try to understand it, but have made no progress. And the other hedgehog and Wnt subtypes don't have that, equal numbers. The single cell data, as I said, there is indeed heterogeneity within tumors. They tend to stay, they're obviously within the same subtype. So the heterogeneity reflects other properties. And the subtype, one of the most accurate ways of defining the subtype now is to use DNA methylation data. So that you get DNA methylation microarrays and get on huge numbers of genes in their methylation state. And it seems to reflect the cell of origin. Once you're in one of these subtypes, you're there, and it kind of reflects where the tumor came from, how it got to where you are. Doesn't tend to change over time. So the heterogeneity within the tumor at a single cell level really reflects the evolution that's occurring at a cellular level within the tumor, and we're just beginning to understand that. As I said already, one of the features is that it can identify a population of stem cells. They're not really stem cells; they're cancer-generating cells. But what other features we'll find, it's not so clear yet. But I think it's an interesting concept. At first I was kind of skeptical, like stem cells versus cancer. It's all cancer, but it turns out that it's really true. The biologic studies show if you transplant the stem cells in, it forms tumors, and if you do the other ones, no tumors. So there really are different populations within the cell.
So I would take the role of asking the last question, and that actually relates to that symptoms all over that where you started. For some Metroblastomas, it seems like it's likely that they do originate from granule cell precursors.
The sonic hedgehog subtype.
The sonic hedgehog. But for the other one, particularly the atypical Rhabdoid.
Yeah, those are not even granules. Those are not even the cerebellum. That's
not even cerebellum. So do you have any sense, particularly because you suggested that one of the things with the state of being in a tumor is basically a differentiated progenitor state.
I believe that, I think for all cancers, not just brain tumors, that there is a component of their oncogenesis is the state of differentiation of the cell of origin at the time that it got the hits that led to the cancer, the genetic hits that led to the cancer.
So my question is, does that state have to be maintained by a signaling medium that is hospitable to maintaining the tumor in its differentiated tumorigenic state? Or do you simply need to maintain the tumor stem cell?
Yeah. So as I said to Albert this morning, I'm an old guy, and I'm hoping that I've written my last grant and don't plan to continue doing. I'm turned 70 next year. I say it out loud, but if I were to start over now and write a grant, and I'm fascinated with the notion of cell state as defined. I mean, we can define it in terms of the epigenome, which genes are turned on, but then you go, well, why? What defines that ensemble of genes as being, and how does that change? What are the signals that lock it into that state? And how stable is it? Are there other ways that you can get into it? I think it has, and Albert and I were kind of wondering, what are the signals when cells contact each other, and and there's a life or death signal that comes from that. Is it, we tend to think of like neurotrophic factors and transcription factors, but there may be, ultimately it boils down to decisions that cell is making about life or death, and its state of differentiation is an important component. And so I kind of feel like trying to understand at a fundamental level what defines and changes states of cells is going to be important for development of the nervous system, understanding nervous system diseases, and as well as cancer. That's.
So, are there any more questions? Dr. Pontine, I guess.
Scott.
If not, let's thank Scott again for